Good morning, everyone. Um, it's a great pleasure to discuss some of the work that I did while I was in IITA from 1979 to 2006. First of all, I want to thank Corteva Agri-Science for organizing this symposium, and Catherine and her group for scheduling this and inviting me for this presentation. The topic is conventional and molecular breeding of cowpea for food and nutritional security in Africa and beyond. And most of this activity was done while I was at IITA from 1979 to 2006. You can see this about 27 years of my life. And it was a pleasure to be there. And my colleagues at IITA and many scientists around the world were very helpful. So I just want to go through briefly a set of slides and explain what has been done and how a crop that was a crawling type grown by many African farmers. And this is a crop that originated in Africa, and I'll be showing the next slide. And it is mainly grown in intercrop with sorghum, millet, and maize with very low density, low fertility, and so on. So the first thing to improve this challenge was to change this plant type into a respectable crop, and using the germplasm variability, crossing erect type with crawling type, we developed very erect type varieties, early maturing. And this system was transformed to this system, which is 300–400% more productive. And towards middle of my work, we noticed that one of the insects, and I'll be talking about that a little bit later, Maruca pod borer, we had no resistance. So we had to bring Bt gene into cowpea, and that's the story I'll be slowly mentioning to you. So cowpea originated in southern part of Africa, and it is now grown in over 100 countries around the world. It is used in many ways. Vegetable cowpea, which is yard-long bean, and it is mainly in Southeast Asia, plus part of Americas. The grain is the major food crop, and it is, here is a major food product from Nigeria and West Africa, and the boiled beans. Also the fodder, it's grown as a fodder crop. In southern and eastern part of Africa, the cowpea leaves are eaten as a spinach. So it's a very versatile crop. It fixes nitrogen, and so this is a kind of fits in many systems. It is grown, as I mentioned, over 80 countries, covering 14.5 million hectares with a production of about 8.5 million tons. Mostly it is grown in West Africa, the major crop in West Africa, the semi-arid region. But recently it has become a major crop in Brazil also. It is grown in southern part of US, southern Europe, and part of Asia. So it's a major crop in mainly West Africa, but throughout the world. In West Africa, I showed this slide earlier, it is mainly grown as an intercrop with millet and sorghum, and in West Africa and in Eastern Southern Africa with the maize. And both grain and fodder are very important. And here is a map of West Africa. This is the dry arid region where cowpea is very important. And the International Institute of Tropical Agriculture, where I was based, is here in Nigeria, about Ibadan. And IIT established a Kano station, IIT Kano station in northern Nigeria, where I was located for many years. And most of the farmers in this region grow the cowpea. This slide is from one of the farmers from this side here. The cowpea research started in early 1900s, primarily in US, and then later on in 60s, systematic breeding programs were started in US, India, Asia, and parts of Africa. But the main thrust on cowpea research started from 1967, when the International Institute of Tropical Agriculture was established at Ibadan, Nigeria. This is one of the three international centers established at that time. The first one was CIMIT in Mexico, and then IRI in Philippines, and third one was IIT in 67. The work on cowpea started from 1970 onwards, and the major focus in first decade was the conservation of collection and conservation of germplasm and evaluation of this for many diseases and insect resistance and so on. I joined here about 79, and since then up to 2006, I retired, I was there. Plus, I'm still continuing on cowpea research as a retired person, partly in India and US and consulting in Brazil. Now I'll be showing some slides on that. So the major focus in early part was germplasm collection and changing the plant type and seed type to 60-day early maturing, disease and insect resistant, including the parasitic weeds, Striga, Electra, and improved cropping systems, and subsequently for low phosphorus, heat tolerant protein and iron. And it was here that we found problem with the Maruca part borer, for which there was no resistance. And so we requested molecular breeding programs, and this is the story I'll be telling a little bit later. So cowpea is one of the food legumes which has minimum anti-nutritional factors, both in leaves and seeds and so on. So it is preferred by everything, all the pathogens, insects, including human beings, cattle, and everyone likes cowpea. So there are many diseases, about 30, but about 15 diseases by bacteria, fungi, viruses, and nematodes. Some of these I have listed here. I don't want to go through these. Many diseases and major 5 insects, particularly in Africa: aphids, thrips, Maruca part borer. This is one of the insects I'll be talking a little bit more. Pod-sucking bugs and storage bruchids. In Western Hemisphere, in US, there is curculio and some other pests that are not here, but mainly in Africa, these are the pests. 2 parasitic weeds, Striga and Electra, and abiotic constraints since it is in dry, semi-dry regions, heat and drought and low fertility. And farmers use the spreading type, so it is spreading late maturity and low yield potential. So if the grain and fodder both are important, if you cut 1 square meter here, you will get very little grain and fodder. So these are the constraints that we started in the beginning of the program. And I'll quickly go through just a few visuals of these diseases, how devastating they can be. So many bacterial diseases, fungal diseases, I have listed a few of them here, and then this is the nematode. So these are some of the diseases that were major. Viruses, there are 8 major viruses. I have listed 5 of them here, and they are all very important in many parts of, and this thing is more in the wetter region, this is more in the dry region. So both wet and dry regions of Africa, viruses are there. And these are the Striga, parasitic Striga, those who have not seen this. This is Striga gassneritis. The seeds are in the ground, and when the cowpea germinates, the seeds of Striga germinate and attach to the roots of cowpea, and then they start sucking the sap from the plant, and the plants die. There is another parasitic weed called Electra, and in wetter region particularly, but slightly higher rainfall, this is a major problem, and when it attacks, almost 100% yield loss. So these are the 2 parasitic weed. These are only in Africa. It is not; these are not in other parts of the world. And then the insects, aphids are the major. Apis craxivora are the major insects, and I have this slide is from Texas A&M where I am here. So the same aphid is in Africa as well as in other parts of the world. And thrips, flower thrips, they attack in the emerging buds of the plants, and they will drop all the buds. The buds will not develop, the plant will look less green, but there will be no flower and pod. And this is the insect, the thrip. And this is the Maruca part borer. It's a moth. It lays eggs in the base of the flower bud, and the larvae will hatch, and this will attack the buds, the flowers, as well as the pods. And this is the larvae in the pod, and a mature pod looks like this. And the damage can be 80 to 100%. This is one of the major pests, and I'll be talking a little bit more about this as we go along. And pod bugs, after the Maruca have done the damage and some pods are there, These are the number of pod bugs that come and suck the sap, and then the pods will prematurely dry out. And whatever has been left goes to farmer's home, then this bruchid will come and attack. And after 2-3 months of storage, the susceptible varieties become almost worthless. They're not even fit for animal consumption. So there is a relay race of insects from aphid, thrips, maruca, and then the bruchids. And since it is grown in the semi-arid regions, drought is one of the major problems. This is a slide from Niger Republic, just north of Nigeria. And this is a millet and cowpea intercropping. Here you can see the millet plants are almost dying and cowpea is virtually surviving. So drought can be one of the major problems, drought and heat. And heat is another one. This is a pot grown behind my building in Kano, Nigeria, in month of April, May, June, when temperatures go very high. So 41 degrees Celsius, the susceptible cowpea will drop, all the flowers will almost look like sterile, and the heat tolerant line is this. Same thing in the Texas A&M, we have seen here, drought heat susceptible line, and this is low phosphorus tolerant. So there is a field here which is very low in phosphorus and nitrogen, potash, and we screen line. There are some which are good, some which are very bad. So drought, heat, and low phosphorus, low fertility are the major abiotic constraints. So as I mentioned earlier, the first decade in IITA collected about 15,000 germplasm lines, including some wild types. And when I joined IITA, these lines were being screened for sources of resistance insect. And my main focus was plant type. So we looked at the germplasm, and there were plants which, these are planted the same day, 155 days maturity and 55 days maturity. And the plant type was from crawling to erect, very early maturing, and the tall peduncles to, so this was like a paradise for a plant breeder. So we had so many diseases and pests, insects, abiotic and biotic species, but germplasm offered us an opportunity to improve the crop. And since early maturity is 55 days, we can move very fast, 3-4 cycles in a year. So we capitalized on this and we screened the germplasm for the diseases, insects, and pests. And I'll just quickly go through this. So we found resistance to all the 8 viruses, all the major fungal and bacterial diseases, including nematodes. So we got very good sources of resistance, including Striga. So there was a germplasm line from Botswana, B301, which was resistant to Striga. This is B301. This is Striga susceptible. This is an F1, it was one dominant gene. Same thing for Electra. The same B301 was also resistant to Electra. And both of these were resistant, highly resistant. Here is a pot, in the same pot, the 3 plants. And the Striga resistance was kind of hypersensitive reaction. So when the seed will germinate, attach the susceptible plant, it will develop the haustoria and the plant will remerge. But on resistant plant, the cell will die. It will be a hypersensitive reaction and it will look like this, completely resistant. One dominant gene, and we worked out the inheritance of this. For aphid, we found very good source of resistance, again, one dominant gene. For thrips also, we had very good source of resistance. Here is a thrip susceptible line, next door is thrip resistant line. But for maruca, and we tried to screen in a screen box here, very little resistance. And in the field, there was some level of resistance, but this was mainly due to thrips and little bit damaged by this. But in severe incidence of maruca, there was almost 0 yield. So we had good source of all the diseases, parasitic weeds, thrips, aphid, and also I had good source of resistance to bruchids. It was one of these slides earlier. Then for abiotic stresses, we started a program. This was the first time we started in the world. Bring the drought in the greenhouse. We have a boxes, wooden boxes, and you about 4-inch soil, plant the test line, water them for 2 weeks, and after that stop watering and see which one dies first. So very good differences were there. So so maize and soybean will die within 1 week. Cowpea, some will die early, some will late. So within cowpea, we found some very highly tolerant cowpea lines and highly susceptible cowpea lines. So we had very good source of this I called as a shoot dehydration tolerant. And they were of two types. The tolerant lines will live longer, and some of them will die all of a sudden, but others will drop the lower leaves, but keep the tip alive up to 70, 80 days after terminating the water. Independently, we looked at the root. So screening of root by box method, here is a deep and dense root system versus umbrella type. And we combined this with this and got something like this. So in the field, we tested this. So this is the drought susceptible line. This is the drought tolerant line with deep and dense root system. For same thing for the low fertility, we screen lines in a very poor soil, and we got major variability among the germplasm for the low phosphorus tolerance. And we tested this. This is work done at Texas A&M in the pod culture, and this is the susceptible to phosphorus, and these are the tolerant to phosphorus. And we found again one dominant gene for the low phosphorus tolerance. Some of these lines which have low phosphorus tolerance and deep and drought tolerant genes, a slide from Niger Republic. This is a slide from Niger Republic, and you can see the drought susceptible lines are completely dead while others are doing very well. So we developed good source of resistance to diseases, some major insect pests, drought and heat tolerant. Then the quality also, we screened germplasm line. This was through the Harvest Plus project starting from 2002. And we got the range of protein from 22 to 32%. And very quickly, we combined this to measure forces. Now, as I mentioned earlier, the problem in Africa was the low yielding, spreading type, late maturing type. So the challenge was how to improve the plant type. And we followed the model of the wheat and rice, which had been done earlier. So we crossed the erect type with the spreading type, germplasm had there. And also we took the early maturing germplasm lines and crossed them. So this type of cowpea became this type of cowpea, and we selected for early maturity, and we had a almost revolution in plant cowpea breeding. So from spreading type to 60-day early maturing, 75-day early maturing, and dual purpose. So all kinds of varieties that came. This happened very quickly before 1890, and we developed systematically. When I joined IITA, somewhere around here, most of the lines were susceptible, local line. Ife Brown is one of the major improved variety in Africa and Nigeria. And this was susceptible to most everything, but it was very high yielding. So we use this as a base. And and subsequently over the years, we developed lines which were resistant to everything. And I want you to remember this line, IT97K 499-35. This was developed in 1997, which has resistance to almost everything, but not for Maruca part borer. So we had reached in mid-90s when there was lines, 60-day cowpea maturity, high yielding, resistant to most everything, except for Maruca part borer. And at that time, I had been invited by Purdue University, and I gave a talk, and the talk of, at that time, the discussion on BT was going on. I'll come back to that a little bit later. So just to tell you, to show you, this is Danila, one of the local varieties grown by farmers. The yield was 0.5 ton, and the fodder was almost 3 tons. The new varieties, yield was 2.5 tons, and fodder was almost the same as that one. So we, through the systematic improvement, the yield has become almost 4 or 5 times more compared to what farmers had done. And the protein content, I had mentioned, some of our improved varieties were 30% protein and loaded with iron and zinc compared to other crops. So the quality was also improved. And these lines IIT has a global mandate. It was distributed to over 60 countries around the world, and 40 of these varieties have been released. Here is a list in different regions. I don't want to go through that. And that brought cultivation of cowpea in many areas and very quick increase in the global area. This is one of the slides from Nigeria, where the new variety was shown in the farmer's field. And when we have something good, almost everyone is interested in looking at it. And here is Amir's brother himself with a lot of farmers. You can see a good crop of cowpea. The other thing that happened was this farmer's system of intercropping, we changed it to strip cropping and improved varieties with improved sorghum varieties with minimum inputs. And that revolutionized the cultivation. So this is intercrop sorghum with cowpea. This is sorghum with cowpea with improved method of cultivation, improved variety. And what has happened between here and here is we took this row of sorghum and put it here, took this row of sorghum, put it here. So 2 dense lines of sorghum and 4 lines of cowpea, put fertilizer here and 1 or 2 spray of insecticide for maruca here. And the yield was 400–500% more compared to this system. And this became very popular. And many farmers, this was a project funded by USAID, and USAID was very happy. And even women farmers from Hausa land became partners in this particular thing. And Nigeria was producing 500,000 ton cowpea when I joined. And very quickly, by 2002, 2004, it was almost 3 million tons. So it was a very big success story. But as I mentioned earlier, we didn't have resistance to maruca pod borer. And here was the fact from the field. So some of our improved varieties, you spray 2 times for maruca, you get 2.6, 2.7 ton yield. No spray is 1/5, but it was better than the local variety. Local variety Danila, if you don't spray, you don't get much. So even if you don't spray, the new varieties were giving higher yield and farmers were happy. But you spray 2 times, the yield goes 5 times. So this is where we started collaborating with Purdue University initially, and they promised us that BT gene would be added to our improved varieties. And this is again, I have showed this slide earlier. The maruca moth lays eggs here, the larvae comes and does the damage, and you have a lot of it. So this was one of our challenge. And since BT gene had been already at that time put in some of the crops, and 1996 BT cotton was released. And so that stimulated discussion on how can we now breed for maruca pod borer for cowpea. And that if we eliminate this 2 spray, all the farmers will get 5 times more yield than what they're getting, and there will be a revolution. So this was a dream for all of us, and it worked over time. So as I mentioned earlier, when we had good crop, we bring all the big shots in the field and show them. So here is Amir's brother himself with USAID officials. Here is Amir himself with other officials. And Dr. Borlag himself, he was working with us. He was working on maize and wheat, and he came to our field. And all the big shots came, and we mentioned to them that we are stuck with BT cow, with a maruca resistant thing. And if Monsanto and USAID can help us bring BT gene in cowpea, that will revolutionize cowpea production in Nigeria. And that message clicked to everybody's mind, and people started working along this line. And USAID took the lead through Rockefeller Foundation, and Purdue University was a catalyst activity. IITA was playing a major catalytic role. And these were the partners that got together. IITA, AATF, USAID have listed the whole thing. And Ed Southern, who developed the Southern blotting technique, he had a foundation, and a meeting was organized in 2001 in Dakar for all the partners, and here 5 decisions were taken. Monsanto agreed to donate BT gene royalty-free for Africa. Ed Southern gave funds for 4 countries for biotechnology development. And TJ Higgins is my friend from Australia. He had already developed a pea transgenic pea. So he agreed to put BT gene into cowpea. Rockefeller Foundation and USAID agreed to provide funds. IIT provided the germplasm, improved germplasm, and the real work started in 2004 and 2006. The first BT cowpea was developed by T.J. Higgins lab. Just to tell you that this work was done by IIT and Purdue University. Among the BT proteins, Cry1AB was the most effective to kill the Maruca larvae. So this is a gene that was transferred. So this is a story that multi-partners were involved, and Monsanto was kind enough to donate the gene to Africa royalty-free. And it started in 2001, the discussion here. 2004, the work started. 2006, the first transgenic plant was developed. And here is a publication for that. And it was first put in a variety called IT86D1010, which is resistant to all the viruses, but not very high yielding, but it was giving best regeneration. So T.J. Higgins and group used this one to transform this, and he got several lines at once. So first, when it came, it was tested in Puerto Rico because Africa didn't have the testing protocol for BT gene. And from 2009 onwards, it was tested in Nigeria, Burkina Faso, and Ghana. And here is my colleague, Muhammad Ishaku, who did all the breeding work subsequently, the backcrossing work, and so on. And I mentioned earlier, a variety 97K499-35, which was developed in 97, had resistance to everything except Maruca. So this was used later on to transfer the gene from 1010 to this one. And this was released in 2019 as SAMPI 20T by the Ahmad Bello University under the leadership of Muhammad Ishaq. So this is something that came as a, we had a dream, and the dream came true because of the multi-partner effort. And just to show you the field testing that was done through 1010 as well as 35, there was zero infestation in the BT version, and the non-BT version was susceptible. And so this BT cowpea was tested under the protection leadership of this AATF, African Agricultural Technology Foundation, one that coordinated the whole thing. And this now is 2020, this is a field BT cowpea from the field from Nigeria in 2020. This season, over 2,000 farmers had planted, and hopefully I am expecting that there will be definitely a revolution in cowpea production to start with in Nigeria, and hopefully it will spread to other part of Africa, and we'll see a quantum jump on cowpea production without any insecticide spray. I just want to mention here that BT technology, the GM technology, the anti-GM group has been working hard to confuse people, but Nigerian government, Nigerian people see the benefit for this, and this is a good decision that they took. I'm a plant breeder, and I want to mention this, that this technology, the GM technology, is now more than 25 years old. Millions of farmers are growing it. It is a good technology, and there should be no anti-GM group against particularly some of these in BT and herbicide tolerant which are there. Then this success brought other donors, and I have a friend and colleague, Dr. Stephen Long at University of Illinois, who has got Bill Gates Foundation money to increase photosynthetic efficiency in cowpea. So he has developed a system where the photosynthesis becomes 25–30% more by transgenic work. And in cowpea, we have got major differences in photosynthetic efficiency already. So I have provided him materials, and his group, he has about 8–9 other countries working with him. So a major work is going on improving photosynthetic efficiency in cowpea. Another friend of mine in Australia, Anna Kultinau, and she is a biologist. She has devoted her whole life on apomixis, and she has got another fund from Gates Foundation to bring apomixis in cowpea to fix hybrid cowpea, so that And we have seen hybrid vigor in cowpea. This is one of the slides when I was in Kano, we put it. These two parents were crossed and we get almost 50% heterosis. So she's trying to bring apomixis in cowpea so that the hybrid can be fixed and farmers can save the seed. And my colleagues in Texas A&M are already mapping heat and drought and aphid tolerant genes, plus increasing the protein content through gene editing and other kind of work that is going on. So the cowpea, which was an African crop, not much interest was done, is now had got a global support. And I'm very hopeful that Nigerians and other people working on cowpea will continue work on this. And this is a major, there is a book that I wrote some time back, cowpea will emerge as a major food legume in the world in the coming years. Now, after retirement from Nigeria, I made an arrangement to be a visiting professor at Texas A&M and also in India, and I collaborate with my Brazilian colleagues. So this is the work that we have done in Texas A&M here. It is heat tolerant, drought tolerant, and low phosphorus tolerant. And the heat tolerant gene is 1QTR. It is now the gene is being tracked. Another thing we found that these 70-day maturity was crossed to about 80-day maturity. And in real slime, we got something that matures in 45 days. So we have now cowpea lines, which we call 50-day cowpea lines. And this, so there is an opportunity to reduce the maturity and also increase the yield as we go along. So my work in India, I will briefly go through this for a minute or so. So we grow in northern India wheat and rice. And in between summer, there is 2.5 months place where there is nothing grown. So I'm introducing 60-day cowpea between wheat and rice to grow 3 crops where only 2 you are growing. And we have introduced the varieties directly from Nigeria, plus now breeding there. We have released six varieties now, 5 are listed here, which are being popularized. And in Brazil, cowpea grown in the northeast Brazil through 60-day cowpea that we introduced in Brazil, and Brazilian scientists have worked on it, that in the middle part of Brazil, which was mainly soybean country, they're double cropping. After soybean, they plant a crop of cowpea, and Brazil is now the 3rd largest producer of cowpea in the world. And they have developed a complete mechanization system. So you can see the soybean was harvested, and these are the soybean residues. The cowpea is being planted, cowpea germinates among the soybean residue, and this is a good crop of cowpea, and it is combined. And so cowpea is now being exported from Brazil to India and some other countries. In US, it's a system where wheat is grown from October to almost May, June, and then left fallow. So in between here, we are trying to get a double cropping, the 60-day cowpea here, and then one. So this system comes, here is a field trial from Vernon in Texas, where the wheat was harvested and cowpea was planted, and we got very good yield. So there is a potential. This is what we are doing in Texas A&M, very good potential. The varieties that we brought from Nigeria, resistance to everything, were crossed to US varieties, and we got some plant types which are even better than what we had in Nigeria. So these are some of the lines that we have developed here, and this is how they look in the field, about 2.5 to 3 tons per hectare in 60 days. Actually, they mature here much earlier than in Africa, and hopefully next year we'll be releasing some varieties here. We had a project from Buffett Foundation for South Africa, and these are some of the new varieties developed in Texas A&M. We tested there, and they look like miracle. You can see that these are my colleagues from University of Limpopo that we are testing at Ukalima. And these 2 of these varieties were released in South Africa. This is Texas A&M Cowpea 1 and Texas A&M Cowpea 2. This is a slide from Ukalima farm, seed multiplication farm. You can see it is like wheat and rice, 3 tons per hectare. And the cowpea production from 1960, I joined in 81, but in 60, the production has become 400–500% more, almost more than wheat and rice. So overall, and with the BT cowpea released in Africa, many countries growing, I'm very hopeful that the production will go much more as we go along. So this is a book that Crop Science Society of America published in 2014. The title is The Food Legume of the 21st Century. So with resistance to all the major diseases, maturity from 50 to 60 days, fitting into cropping systems, and in Africa, Asia, Brazil, in America, everywhere, we are very hopeful that cowpea will be a major food legume in the coming century. This work was done by many people in IITA, and I'm acknowledging these colleagues in IITA, Pant University, Texas A&M, Purdue University, and all other major foundations. In Texas A&M, we had many professors and students who contributed, and overall, my wife and family have been all together for these years that I have been. Just before I end up, I want to say that for plant breeders, the young plant breeders, you take a crop and put your mind and life into it, and you'll come up with something that you can feel good about it. And I feel very good now that cowpea is becoming popular. And I want to thank again Catherine and her group, and for inviting me and giving me this opportunity to share this work with you. And I hope that African agriculture will infuse more technological and also add more fertilizer and inputs so that its production can increase over time and keep pace with the population growth. So thank you very much, and I will be glad to answer some questions.